Imagine a microscopic instruction manual, intricately coiled within the nucleus of every cell in your body. This is DNA, the blueprint of life. From the color of your eyes to your predisposition to certain diseases, DNA dictates the traits that make you uniquely you. DNA's story is one of scientific discovery, groundbreaking research, and the unraveling of the very essence of what makes us human. In 1953, James Watson and Francis Crick, with the help of Rosalind Franklin's X-ray diffraction images, unveiled the double helix structure of DNA. This revelation revolutionized biology, providing scientists with the key to understanding how genetic information is stored, replicated, and passed down through generations. DNA's power lies in its ability to store and transmit genetic information. Picture a vast library filled with countless books, each containing the instructions for building and maintaining a living organism. Genes are segments of DNA that code for specific traits. These traits, ranging from physical attributes to complex biological processes, are determined by the sequence of chemical building blocks within DNA. DNA connects us to every living thing on Earth, a testament to the shared ancestry of all life forms. From the smallest bacteria to the largest whales, the language of DNA whispers tales of evolution, adaptation, and the interconnectedness of the natural world. To truly appreciate the elegance of DNA, we must delve into its structure. Imagine a twisted ladder, spiraling gracefully towards the heavens. This is the double helix, the iconic shape of DNA. The two sides of the ladder, known as the sugar-phosphate backbones, provide structural support to the molecule. Connecting these backbones like rungs on a ladder are pairs of nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. They pair up in a specific manner, A always with T and G always with C. During replication, the double helix unwinds, and each strand serves as a template for creating a new complementary strand. Picture a grand spiral staircase, an architectural marvel that winds its way through a towering structure. This is the essence of DNA's double helix, a microscopic marvel that houses the blueprint of life itself. The handrails of this staircase are akin to the sugar-phosphate backbones of DNA. The steps that connect the handrails are analogous to the base pairs that bridge the two strands of DNA. Like a carefully crafted message encoded within the steps of the staircase, the sequence of base pairs dictates the instructions for building and maintaining a living organism. Imagine a language composed of only 4 letters, a seemingly limited alphabet that nonetheless holds the power to convey an infinite array of instructions. This is the language of genes, written in the sequence of base pairs along the DNA double helix. Adenine, thymine, guanine, and cytosine are the 4 letters of this genetic alphabet. This precise sequence determines the genetic makeup of an organism. Like a code waiting to be deciphered, the sequence of base pairs dictates the instructions for everything from the color of your eyes to the shape of your proteins. Imagine a zipper, its interlocking teeth separating with ease as we pull the slider down. This is the essence of DNA replication, a process that allows cells to make faithful copies of their genetic material before cell division. The DNA double helix unwinds, breaking the hydrogen bonds between the base pairs and exposing the 2 strands. Each separated strand serves as a template for the synthesis of a new complementary strand. This process, known as semi-conservative replication, ensures that each new DNA molecule contains 1 original strand and 1 newly synthesized strand. Imagine a grand orchestra, its musicians working in perfect harmony to produce a symphony of sound. This is akin to the intricate interplay of molecules and processes that transform the genetic information encoded within DNA into a living, breathing organism. DNA itself is but the blueprint, the musical score that guides the orchestra of cellular machinery. During transcription, the DNA sequence is copied into a messenger RNA molecule, which carries the genetic information to the ribosomes. At the ribosomes, the mRNA molecule serves as a template for assembling amino acids into a specific sequence, giving rise to the proteins that carry out a vast array of functions within the cell. Imagine a team of skilled architects meticulously poring over a blueprint, translating its intricate designs into a tangible structure. This is the essence of gene expression, the process by which the information encoded within DNA directs the synthesis of proteins. Genes serve as the master architects of this cellular construction project. Each gene contains the code for a specific protein, dictating the precise sequence of amino acids that must be linked together to form a functional molecule. From the initial transcription of DNA into RNA to the final assembly of amino acids into a protein, each step is carefully regulated, ensuring the faithful execution of the genetic instructions. Imagine a scribe meticulously copying a precious manuscript, ensuring that every word, every punctuation mark is faithfully reproduced. This is the essence of transcription, the first step in the journey from gene to protein. During transcription, the DNA sequence of a gene is copied into a messenger RNA, or mRNA molecule. This molecular copying process is carried out by an enzyme called RNA polymerase, which binds to a specific region of DNA at the beginning of a gene. Once the entire gene has been transcribed, the newly synthesized mRNA molecule detaches from the DNA, carrying with it a faithful copy of the genetic instructions. Imagine a skilled translator deciphering an ancient text, converting its cryptic symbols into a language that can be understood. This is the essence of translation, the second step in the journey from gene to protein. During translation, the mRNA molecule is transported from the nucleus to the ribosomes, the protein synthesis machinery of the cell. Ribosomes act as molecular interpreters, deciphering the mRNA code and using it to assemble amino acids into a specific sequence. Transfer RNA or tRNA molecules recognize specific codons on the mRNA and deliver the corresponding amino acids to the ribosome, forming a growing polypeptide chain. Imagine a grand symphony, its intricate melodies and harmonies arising from the coordinated efforts of countless musicians playing in perfect synchrony. This is akin to the symphony of life, the intricate interplay of genes, proteins, and cellular processes that gives rise to the complexity and diversity of living organisms. Genes direct the synthesis of proteins, the musicians that carry out the myriad functions essential for life. The precise orchestration of gene expression is essential for the development, growth, and maintenance of all living organisms. This intricate dance of molecules highlights the profound connection between our genes and the remarkable tapestry of life that surrounds us. Imagine a dusty attic filled with forgotten treasures, each holding a clue to a larger mystery. This is akin to the world of DNA research in the early 20th century, a time when scientists were just beginning to unravel the secrets of this enigmatic molecule. The discovery of DNA itself, back in 1869 by Swiss chemist Friedrich Miescher, was but the first step in a long journey to decipher the language of life. Landmark discoveries, like Oswald Avery's identification of DNA as the carrier of genetic information in 1944, and Erwin Chargaff's observation of base pairing rules in 1950, laid the groundwork for the revelation of DNA's double helix structure by Watson and Crick in 1953. This revelation marked the beginning of a new era in genetic research, opening up a world of possibilities for understanding and manipulating the building blocks of life. Imagine a global relay race with scientists from around the world passing the baton of knowledge, each team contributing to a monumental goal, mapping the entire human genome. This was the essence of the Human Genome Project, a collaborative international effort launched in 1990 that aimed to determine the sequence of the 3 billion base pairs that make up human DNA. This ambitious project required the combined expertise of geneticists, computer scientists, engineers, and countless others. The completion of the Human Genome Project in 2003, marked by the publication of the first draft of the Human Genome Sequence, was a watershed moment in human history. This monumental achievement opened up new avenues for understanding human health, disease, and evolution, paving the way for personalized medicine, gene therapy, and other groundbreaking applications. Imagine a microscopic pair of scissors, capable of precisely snipping and editing the very fabric of our genes. This is the essence of CRISPR-Cas9, a revolutionary gene editing tool that has taken the scientific world by storm. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a natural defense mechanism found in bacteria. The CRISPR-Cas9 system acts like a molecular surgeon, guided by a short RNA molecule that recognizes a specific DNA sequence. This groundbreaking technology has revolutionized genetic research, offering the potential to cure genetic diseases, develop new therapies, and even enhance human capabilities. Imagine a vast, uncharted territory, filled with both promise and peril. This is the ethical landscape of DNA research, a frontier where scientific breakthroughs are forcing us to confront profound questions about the very nature of life, identity, and our responsibility to future generations. As our understanding of DNA deepens, so too does our ability to manipulate it, raising ethical dilemmas that demand careful consideration. The power to edit the human germline, for example, raises concerns about unintended consequences, designer babies, and the potential for exacerbating social inequalities. The use of genetic information in areas like insurance and employment raises concerns about discrimination and privacy. These are complex issues with no easy answers, requiring thoughtful dialogue between scientists, ethicists, policymakers, and the public. As we venture further into the uncharted territories of DNA, we must proceed with caution, guided by a strong ethical compass. Open and transparent discussions about the potential benefits and risks of DNA technology are crucial to ensuring that these powerful tools are used responsibly for the betterment of humanity and the preservation of the delicate web of life on Earth. Imagine a world where medical treatments are tailored to your unique genetic makeup. This is the promise of personalized medicine, fueled by our expanding knowledge of DNA. By analyzing genomes, scientists can identify genetic variations influencing disease susceptibility and medication response. This approach is transforming diagnosis, treatment, and prevention. Genetic testing can identify cancer risks, enabling early detection and targeted therapies. Pharmacogenomics helps doctors prescribe effective medications with fewer side effects. Personalized medicine promises more effective, proactive, and individualized healthcare. Imagine embarking on a journey, not through physical landscapes, but through the intricate pathways of your own DNA, tracing the footsteps of your ancestors across time and continents. This is the allure of genetic ancestry testing, a rapidly growing field that leverages the power of DNA to uncover our ancestral roots, connect us with distant relatives, and shed light on the migrations and interminglings that have shaped humanity's tapestry. By analyzing specific markers within our DNA, scientists can trace our lineage back thousands of years, revealing the geographic origins of our ancestors, their likely migration patterns, and even the potential intermixing with other populations. Companies like 23andMe and Ancestry DNA offer direct-to-consumer genetic ancestry testing kits, allowing individuals to explore their genetic heritage from the comfort of their own homes. Beyond satisfying our curiosity about our family history, genetic ancestry testing has implications for fields like anthropology, history, and even medicine. By mapping the genetic diversity of human populations, scientists can gain insights into human evolution, migration patterns, and the genetic basis of certain diseases that are more prevalent in specific populations. Imagine a world where crops are engineered to withstand harsh climates, resist pests and diseases, and produce higher yields, addressing food security concerns for a growing global population. This is the potential of genetic engineering, a field that harnesses the power of DNA technology to modify the genetic makeup of organisms, introducing desirable traits and enhancing their characteristics. Genetically modified, or GM crops, for example, have been engineered to resist herbicides, allowing farmers to control weeds, more effectively reduce pesticide use, and potentially increase crop yields. Other GM crops are engineered to produce higher levels of essential nutrients, offering a potential solution to malnutrition in developing countries. Beyond agriculture, genetic engineering is revolutionizing fields like medicine, biotechnology, and even environmental science. Scientists are exploring the use of genetically modified bacteria to produce biofuels, clean up pollutants, and even deliver drugs with greater precision. The potential applications of genetic engineering are vast, offering both challenges and opportunities as we navigate the ethical and ecological considerations of manipulating the building blocks of life. Imagine a future shaped by our ever-deepening understanding of DNA, a future where the secrets of life itself are increasingly unlocked, offering unprecedented opportunities to improve human health, address global challenges, and reshape our relationship with the natural world. This is the promise and the peril of the DNA revolution, a transformative period in human history marked by both extraordinary scientific advancements and profound ethical dilemmas. As our ability to read, analyze, and even manipulate DNA continues to advance at an astonishing pace, we are entering a new era of scientific exploration and discovery. The potential benefits of this knowledge are immense, offering hope for curing genetic diseases, developing personalized therapies, enhancing agricultural productivity, and even unraveling the mysteries of human evolution. However, this power to reshape the building blocks of life comes with a profound responsibility. The ethical considerations surrounding DNA technology are complex and multifaceted, demanding careful consideration and open dialogue among scientists, policymakers, ethicists, and the public. As we venture further into the uncharted territories of DNA, we must proceed with a deep sense of humility, recognizing the interconnectedness of all life and striving to harness the power of this knowledge for the betterment of humanity and the preservation of our planet. We've journeyed from the grand expanse of the cosmos to the intricate inner workings of the cell. At the heart of this journey lies DNA, the molecule that underpins life on Earth. From single-celled organisms to the human brain, DNA's structure serves as a universal language, a testament to the shared ancestry of all living things. Just as the stars have captivated us, so too has the microscopic world within us, revealing wonders that continue to inspire. DNA, once a mystery, is now a beacon of understanding, illuminating heredity and evolution. It's a story told in base pairs, a symphony of genetic information, connecting us to a lineage billions of years old. The realization that all living organisms share this blueprint is awe-inspiring. It speaks to the unity of life on Earth, highlighting the interconnectedness of ecosystems, the balance of nature, and our responsibility to safeguard biodiversity. The journey of DNA discovery is far from over. We stand at the cusp of a new era in genomics, where our ever-deepening understanding of DNA promises to revolutionize fields as diverse as medicine, agriculture, and even space exploration. The tools of genetic analysis are becoming increasingly sophisticated, allowing us to decipher the complexities of the genome with greater precision and speed than ever before. Imagine a future where diseases are diagnosed before symptoms even arise, where personalized therapies are tailored to an individual's genetic makeup, and where the scourge of inherited disorders is eradicated through gene editing. This is the potential of DNA technology, a future where scientific breakthroughs hold the promise of alleviating suffering, extending lifespans, and improving the human condition in profound ways. Beyond medicine, DNA technology is poised to transform the way we feed a growing global population, develop sustainable energy sources, and even search for life beyond Earth. Genetically engineered crops with enhanced yields and resilience to climate change could help address food security concerns, while biofuels produced from algae could offer a renewable alternative to fossil fuels. And as we venture further into the cosmos, DNA sequencing will be an invaluable tool in the search for extraterrestrial life, helping us identify and understand the potential diversity of life in the universe. With great power comes great responsibility. Unlocking DNA's secrets demands caution, guided by ethics and respect for life. Manipulating life's building blocks raises profound ethical questions. Today's DNA decisions shape humanity's future. Open discussions ensure responsible use of genomic tools. Diverse voices must navigate this genomic frontier. The future is written in today's choices. Unravel DNA's mysteries with wisdom and humility. Our legacy is judged by our wisdom and compassion.